Great. Thank you all very much for hanging in there for the last panel. Thank you for inviting me to participate in this important and timely discussion. First thing I do have to do, given my position, is let you know that my views I express here are those of my own personal views on these issues and questions and do not necessarily represent any institution or society to which I'm affiliated. My views on these topics are based on my now more than 25 years, I am old, in translational and genetics, genomics and genetics research. In our studies, we've enrolled several thousands of individuals in genetics research studies and returned well over 2,000 individual test results to those participants. So we've done this quite a bit. My research lab has adopted the approach of embedding within it a CLIA-compliant clinical testing laboratory, and that is both for primary variants that we discover in the part of our genetic etiologic research, as well as secondary findings, as well as also returning results to individuals really as part of clinical experiments. That is, we are generating results that we give to people in order to understand the process of returning genetic test results. We decided to embed the lab within the research lab primarily because of cost. It is much less expensive for us, as has been mentioned by a number of people, to do the CLIA within our own group, within our own laboratory, than to farm that out. The second reason is that it is really increasingly necessary for us. It's part of our, we think it's an important part of our obligation to our research participants to help them benefit from the fruits of this research, as well as to increasingly use genetic test results for our molecularly targeted therapeutic protocols. So we're actually doing precision therapeutics based on the variants we are detecting. I would also mention, because it has come up a few times vis-a-vis costs and other issues, and Susan has raised the issue of the fuzzy boundary between primary and secondary findings, that we have also inaugurated what we're calling a secondary genomic findings service that identifies pathogenic variants from research exomes, validates them, returns them to participants with medical and genetic counseling, and we estimate the cost of that to be about $40 per individual sequenced. That's amortized across all comers, and it's $40 because we are spreading that cost amongst everyone that's sequenced, not just the people that receive the results. We've also returned innumerable primary variants. We've learned a lot from these experiences. I think this has come up a few times. It's been a bit of a genomics-focused discussion, and I think the answer to these issues is very different for different kinds of analytes and tests. But in genomics, at least, I've been impressed that the analytic validity, which is primarily what CLIA is about, is one of the things we should be least worried about in clinical genomics, following on the comments that Rex made earlier. Sequencing, whether it's next-gen or Sanger, is an established methodology. The error rate with reasonable standards in use in most research laboratories is low. That is not the challenge. The challenge in genetic and genomic testing is the clinical validity of the findings, which unfortunately CLIA doesn't have all that much to say about. Research sequencing is necessarily done at the cutting edge of knowledge. And because of that, there is going to be a great deal of uncertainty of the clinical validity of a variant in a gene. And that is a very different thing than clinical testing, where the relationship between variants and phenotype is much better understood. So ironically, that is an issue more for primary research findings than it is for secondary findings, because in the latter, that gene variant phenotype relationship is much better established. The clinical utility question is even more complicated. Some people refer to that as actionability. I prefer to call it clinical utility. It is an extension of the uncertainties associated with clinical validity. And there's a wide range of things to think about, and we actually use that in our group as a bit of a measure for our confidence in findings and our willingness to return them. We often ask the question, what is this going to be used for, and is it reasonable to use this result given all the uncertainties for that outcome? It's also important to be clear in all these discussions about analytic validity, clinical validity, and clinical utility. When you're thinking about the accuracy of a result, all such testing, and this was made, I think this point was made by Fran this morning, all testing is associated with uncertainty. As a panel, you are going to be faced with decisions and you are going to be uncertain about what recommendations to make. And I'm here to tell you that we are all going to make errors no matter what we do. And we just have to take a very clear-eyed and cold approach to this and say, if I do this, what error will I make and what is the consequence of that error? And if I do that, what error will I make and what's the consequence of that error? And if I do nothing, what is the error I will make? Because the passive approach is an error as well, and we need to be very cognizant of that and decide which of these error pathways are more tolerable in which situation. I think it's important to remember as well in this mode of uncertainty and errors. You can set the bar at different levels for different kinds of inaccuracy or uncertainty. And again, you'll make mistakes either way. There are certainly costs and burdens associated with the clinical validation of research testing, but it's also very important to remember that if you want to set the bar extremely high, you are going to pay another large price, which is that fewer people will get results or you will delay the receipt of results for those people. That's another error mode. It has costs. It's the same decision you make when you decide how much safety data you need for a drug before you're going to approve a drug. Earlier approval has one kind of error, later approval has a different kind of error. And I think in many of these examples, what we have to remember is that the perfect really is here the enemy of the good, because an uncertain but partially valid result can be used to make a proper clinical decision for a research participant in their care. So CLIA, I think, is an example of the good, not the perfect. It's far from ideal, doesn't guarantee anything. I will say that one thing it does do for us is it formalizes the process and makes it clear for us when we change from our researcher hat to our clinician hat. We say, okay, now this is a test. Where's the order? Now this is a test. Where's the result? Now I'm reviewing this test as an individual test result, not as something I want to publish in a paper. And who are the two people that are going to sign that report that we're going to send and put in the chart? And just that formal thing, even if all of that analytic validity stuff doesn't do us any good at all, just that process of thinking about the test that way, I think is highly beneficial. And we've learned a lot from that. And if you were to wipe CLIA off the books today, we wouldn't stop doing those things because we think they're so valuable in helping us to make sure that we are transparent and clear about the role that we're playing when we're researchers or acting as clinicians, when it's the right thing to do, and again, what error do we want to make. So I'll stop there. Thanks.